Yeah, thanks. So I'm John Coddington. I'm actually the head of science of the Smithsonian's Natural History Museum. So I come from a museum community, which means we do collection-based research on, and have been doing it, of course, for hundreds of years. And as Neil just said, we're very interested in the potential of collections for genomic research. And in a sense, you guys are studying, here mainly people are doing prokaryote diversity, but so it's interesting how these two communities may get together. And I'm also representing, in particular, a formal initiative from the Smithsonian Institution called the Global Genome Initiative. And it's born sort of two realizations. First of all, that essentially nobody has life on ice, which is to say all the genomes of Earth in biorepositories where you can constantly go back and access a genome that's been studied. So both the vouchers for the genome and also the phenotype that genome produces, which is to say a specimen in a classical museum sense, like a prepared skin or bird or butterfly or whatever it may be. Those are important things to have. So that's our mission, preserving the genomic diversity of life on Earth. We intend to achieve that by building a network of biorepositories, which are basically tank farms, freezer farms. Those assets are just beginning to exist in museums around the world, and it's interesting to realize that for the last, say, 40 years of genomic research, by and large, nobody was putting they might be putting that tissue in a freezer, plugged into plugged into line current in the back of the biology building, but nobody's transferring those genomes and phenotypes to institutions that have as part of their mission professional perpetual care, which is what museums do. So museums need to molt to become museums of genomes, the same way they've been museums of other things. So there's a research and collecting agenda. We can't obviously preserve, I think, realistically, the genomes of all life on Earth, but we could get 50% of the major branches of life on Earth, as well as what you guys are doing here, which is sampling ecosystems and maintaining samples of those, particularly for prokaryote diversity. So why is this important? Just to illustrate what genomics is doing to, say, biodiversity science. These are called dark taxa. I think Chris Meyer from the Smithsonian will be talking about this later on. But what it means is blue means that it's a sequence that was deposited in GenBank with a Latin name, with a binomial attached to it. Pink means not. So in invertebrates, you might expect that pattern. But if you look at mammals, there's an enormous percentage of things that are not named. So these are called dark taxa. They basically, when you blast against them, they come back as uncultured eukaryote. That's a problem. We need to have names of things that are in GenBank. So why is this feasible? This is a brief primer on the structure of biodiversity as we know it, sort of arrayed against the Linnaean hierarchy. Let's just skip down to the level of families. From prokaryotes to primates, there's only 8,000 families of life on Earth. And when you think about it, finding a new family of life on Earth, which is to say I walk out in the woods, pick up a bug, look at it and say, that's a new family, that happens for all of life maybe 2 or 3 times a year. So in effect, our knowledge of the structure of biodiversity at, say, the family level, which means really big branches, is essentially done. We have explored Earth at that level. At the species level, it's going to be really hard. So the interesting question is something that have about the rank of a genus. On average, genera contain about 10 species. So our proposal is essentially to decimate biodiversity. We're going to collect every 10th species on Earth. And there's only about 160,000 of those described. Of course, there are plenty of genera that are still unknown. Prokaryotic diversity is kind of a thing unto itself. But 160,000 things as a major global program in 5 years is not hard. Our museum, for example, accessions about 500,000 specimens a year. So it's not unreasonable. And just to note, in GenBank, there's, without even trying, as of last year, about 6,600 families of life on Earth were already in GenBank, and about 50,000 genera. So without even trying, somebody went out, got some tissue, did something vaguely genomic with it, and which is to say this is not as wildly unreasonable as it might sound. The other thing that's good about biodiversity is that it's actually quite structured and concentrated in this sense. It's sort of genera and families geographically. And this is my colleague Biff Birmingham, who didn't quite make it in time to give his talk. He's hosting the Smithsonian Global Earth Observatories. This was actually a system of 50-hectare plots around the world, about 47 plots in 21 countries, invented really by forest ecologists. And what they did was they tagged every woody plant stem in the plots that was bigger than about a centimeter DBH. So a plot in Panama might have, say, 1 million stems in it in 50 hectares. All of those stems are individually known as individuals, and we know what species there are. So that infrastructure has been going for about 30 years, and we've just recently ramped it up to try to expand that asset for sciences other than forest ecology. So, I mean, consider the platform that gives you to study genomics, right? The plants are the producers of the ecosystem. So you can study the fungi on the plants, you can study the fungi in the soil, you can study, you know, the insects that eat the plants, you can study the predators that eat the insects that eat the plants. All of this in an extremely contextualized place where you know what that plant is, and you know its history, you know how much it grew, and of course you're getting all the abiotic data as well. So this network of sites, and in terms of, say, wanting to have the genomes of all trees on Earth, they have about 4,000 genera. The problem is we don't have a definition of tree. The best one I've heard is a tree hurts when you run into it. But it's about 40% of the world total has already been inventoried in these plots. So putting those genomes on ice and making them available for genomic science is also a relatively feasible thing to do. So, however, back to the biorepository, we have a network of the Global Genome Biodiversity Network, which these are people who have, you know, industrial scale biorepositories, which means they're institutional commitments. It's not just your freezer, you know, plugged into the basement somewhere. And we have that many part of our network around the world. What we're attempting to do is to make a global seamless portal to the tissue collections, basically, for genomics around the world. So there's one portal where you can figure out who's got what are the rules for access, you know, what can I do with it. And Dawn showed up in my office about a year ago talking about these global Earth observatories, and I said, well, I'll give you 10,000 slots in the Smithsonian BioRepository, which has just come online. It's a large biorepository, 24 liquid nitrogen tanks and 58 freezers. The total capacity there is about 4 to 5 million 2 mil slots. So that the marginal cost of doing that institutionally was only about $5 million. So, which isn't much, really, when you think about what we're getting there. There's only 2 million described species of life on Earth. So offering micro B3 10,000 slots in this biorepository for you guys to deposit probably samples of prokaryote diversity there. We're interested, as Neil said, in collecting that because we think that's what we do. You know, I'm interested in having mud samples or whatever it may be that contain genomes that represent the prokaryotic diversity of life on Earth. So we're trying to get museums to follow along with us and build these kind of facilities so that we can begin to archive some of the legacy of tissues that are already in collections around the world, and also, of course, to be ready to archive the stuff that you guys are studying. So I think that's it. These are our 2 little web pages. We're open for business. If you have samples in your labs that represent any kind of biodiversity that you think ought to be maintained, send us an email and we'll start talking about how to transfer it into a public repository. Thanks.